Helium and hydrogen atoms are floating around in space. Helium says to the hydrogen, I think I lost my electron. Hydrogen says, are you sure? Helium says what? I'm positive. Nicely done. What did 0 say to 8? What did 0 say to 8? Nice belt. Maybe draw it. What kind of animal would you never play video games with? A cheetah. Well done. Okay, let's take a peek at the schedule here for a second. So we're going to finish up muscle physiology today. Then we're going to be into the central nervous system, which effectively is the brain and the spinal cord. We'll have 3 lectures in brain and spinal cord, and then we'll have exam 3. Exam 3 is actually on a Wednesday, not a Monday. So that's a little different than what we've been doing so far. I just want you to kind of make note of that. So it's going to be Wednesday the 30th. Okay? So it's the day before Halloween. And then we have Veterans Day coming up in November. And then we actually have a lecture that's online after Veterans Day. So that week, because actually I'm out of town. Remember I said there's going to be a couple of weeks that I'm out of town. So that's on the syllabus. And then the other online lecture, I don't know how y'all feel about this, but 4 o'clock on Wednesday before Thanksgiving. Y'all want to be here? Yeah, exactly. So that's also scheduled as an online lecture, okay? All right, yeah, we're all on the same page on that one. Okay, so just walking through some syllabus stuff that's coming. Just want to make sure you guys see that. Questions before we get going? Okay. So we're going to shift gears a little bit today. We've talked pretty extensively about how muscle is organized, the microanatomy. We've talked about how it works. We've talked about a tremendous amount of vocabulary. And I kind of poked fun out of it in the last lecture, just meaning if you record yourself and you listen to it, there's a lot of words that are jargon that only we understand because we're in this class. Or you're in the field. But somebody outside of the space, it literally sounds like a foreign language when you hear us describe how muscles contract. There's a few words that are, discoverable, like the and. But otherwise, when you talk about muscle contraction, it's pretty amazing what you're able to string together if you do that exercise that I suggested. So we're going to shift gears a little bit today. If you were looking for Monday's lecture, for whatever reason, YouTube was a little slow on the upload. So it didn't finish uploading until this morning, but it's there now, and some people have taken a look at it already. So I'll start the upload tonight and try to get that going pretty quickly. I'm going to be working on study guides over the weekend to get those posted before next week or at the beginning of next week. So some of you have made that request. So I'll get that. It's on my to-do for the weekend. So if those of you that want to work a little ahead, I actually want to encourage that. So there's nothing wrong with that. Okay. So the gears that we're going to shift to today are a little bit on how we use muscle. So the first concept that we need to go through is looking at the strength of a contraction. Because we talked about not all muscle contraction is exactly the same. You're going to recruit motor units at different volumes and different intensities based upon the activity that you're doing. you're putting in or taking out contact lenses. That's a whole lot different than trying to open a jar, right? Like a fresh jar of jelly or peanut butter or pickled beets or whatever it is. You're exercising in the gym, right? You're taking notes with a pencil or with a stylus on a tablet. It's very different than bouncing a basketball or swinging a baseball bat or throwing a softball. So how do we vary some of these different types of muscle contractions? So there's two big ways that we do this. Number one is called the length-tension relationship. And then number two is the cross-sectional area force relationship. So if we first look at the length-tension relationship, and that's this graph that's shown to your right, we can see this graph on the y-axis. That's the axis over here. This is tension generated. And across the x-axis is sarcomere length. And you have these nice convenient pictures where you can kind of appreciate that this is sort of mid-contraction. This is the end of contraction, and this is an overly stretched state. And you can see that tension is low and peaks when you have the optimal overlap between the thick and the thin filaments. So the maximum amount of myosin heads are actually finding the maximum number of binding sites on the actin protein. And if you have the maximum number of cross bridges forming, you get the maximum force that's generated, or the maximum tension. That should make intuitive sense. If you overstretch it and there's not interaction, half of them are able to find the binding site, half of the myosin heads cannot, you're going to get less force. Same thing as if you overcrowd, you're going to get less force. So the maximum peak production of force of a muscle is mid-range contraction. And that's kind of how we are designed. We have a range of motion around joints, and there's muscles with muscle bellies that pull across that joint, and it's optimized to have the peak force production mid-stroke. That's the biomechanics. Okay, so that's the first kind of piece of physics that we want to talk about. The next piece of physics is pretty easy as well, that area is proportional to the force. So the greater the cross-sectional area of the muscle, the greater the force production. In other words, the larger the muscle, the greater the force production. But it's really more of the cross-sectional area of the muscle. So it's not the larger the muscle as a cylinder, it's the larger the cross-sectional area across the main contractile range where you're going to get maximal force. And that's why we have muscles that kind of bulge in certain athletes, because that is the cross-sectional area that makes the most sense to have the maximal overlap across that particular range of motion. Okay? Otherwise, our arms, arms of elite athletes wouldn't have bulges where the muscles are. They'd be one giant cylinder that all the way down. Right? But because we have shape to the muscles, the thickest portion of the largest cross-sectional area is happening because that's where the sweet spot is for the range of motion, where you're going to get maximum overlap. Okay, the next concept is linking, again, sort of the nerve stimulus to the muscle contraction, like what we've been talking about with the neuromuscular junction. Linking that nerve stimulus to the muscle contraction. So if we talk about muscles contracting, and we talk about this terminology of a twitch of the muscle. So the single firing of an action potential, the single firing of an electrical signal, and we're going to unpack that in more detail. We're going to deep dive into action potentials in nerve physiology. Right now, we've defined what they are as an electrical signal, and we'll dissect those and get deeper into the mechanism of action potential in nerve physiology. But right now, what we've said is the action potential is the electrical signal that travels down the nerve And that single signal, bless you, is going to lead to a single muscle twitch or a single firing. You can actually register this electrically. You can see the stimulus to the nerve. So 1, 2, 3, 4, 5, all the way to 9. There's a threshold, and it may be a little challenging to see. Let me see if I can dim the lights to make this a little more obvious. That's obviously not the right switch. Neither is that one. There we go. And number 1 stimulus is corresponding to this muscle fiber, this nerve fiber bundle here. Number 2 is this nerve fiber bundle here. Number 3, so on and so forth. The corresponding nerve fiber is below the individual stimulus. Y'all follow? So in the first one, the electrical signal is too small to reach threshold, so you actually never send the full action potential. And so you can appreciate here, hopefully, in the very back, might be more clear on your screens, that there is not a single nerve that lights up in 1, 2, but in 3, we hit the threshold, we send the signal, and this one nerve fiber that's colored red right in the middle actually is lighting up. And you can see number 4, 2 of them, 5, all the way to 9, you get the maximum voltage signal. It's recruiting all of the nerves of that fiber to fire. Now, the below graph, the bottom graph, is the result of the muscle, because this is a nerve-muscle relationship. We focused a lot on the muscle. This is the most we've talked about the nerve, but we have a neuromuscular junction. We have a nerve-muscle relationship. And so on the bottom, we're looking at the tension that's produced. So hopefully you can now sort of see this relationship in 1 and 2, where no nerves are actually excited or firing because you never reach the threshold to fire the action potential. You have no tension produced in the muscle. Electrical signal never made it to threshold to trigger a full action potential, so you never got any muscle tension, no contraction. 3, you get a little bit of tension. 4, increase till you have maximum contraction of situations 7, 8, 9, where all of the nerve fibers are excited and you're getting the maximum tension being produced in the muscle. Make sense? So you can actually increase the amount of tension in the muscle depending upon how many nerves fire a signal or send a signal to the muscle to say, okay, that's going to be a heavy weight that you're going to need to pick up, or this is a very delicate and light contact lens. So we might fire fewer signals with a delicate light contact lens. We may fire more with a 35-pound dumbbell. How many of you have been, and you're getting a lot of feedback, right? Biofeedback, because you look at the contact case and you're not like, all right, let's do this, right? You look at the contact case and you naturally like, okay, I'm going to be very gentle, right? So you're getting a lot of feedback, visible feedback. How many of you run into the situation where you picked up a package and it tricked you? You thought it was heavier than it really was going to be? That's awkward. Or it's actually heavier than you thought it was going to be. That's also awkward. So that's kind of the way this plays out. Okay, the next side of the slide, we're going to talk about different conditions of the muscle and how they fire or how they contract. So up top, we have what we refer to on the upper left or kind of mid-top, a twitch. So you can see the stimulus across the x-axis, and then every peak of the red line is giving you muscle twitches. And the stimulus is firing a twitch. The twitch itself are individual contractions of the muscle as a response to this nerve stimulation. Now, you can have this situation known as treppe. Treppe is this sort of staircase phenomenon on the upper right, where if you time the signals of electrical stimulus closer and closer together, you actually get an increasing muscle twitch because you don't allow the twitch to relax when you fire the next signal. So you get the staircase phenomenon where you increase trepe to produce a larger contraction event. In the bottom signals, you can see that these dashed lines are the nerve signals across the bottom on all of these. Here you can appreciate that the trepe is so close together that you get this staircase phenomenon known as incomplete tetanus, or an incomplete tetanus, in contrast to the right, where you have complete tetanus. So an incomplete tetanus is like this fluttering of the muscle, sustained fluttering contractions. The whole goal is to increase the tension in the muscle to be able to have greater force being produced at any one time. However, it's not fully sustainable. There is a limit. So you can see that these electrical stimuli are the closest together of all of the ones that we've looked at. You can see that the incomplete tetanus, the trepe effect, incomplete tetanus wears off to a complete tetanus, and then it leads to muscle fatigue, where you actually wear out. And that's where the muscle essentially doesn't fail in the sense that it breaks or snaps, but you can't maintain the contraction anymore. You drop the weight, okay? You can't support it anymore. Whatever you're holding, you can't keep up. So as we go through life, we have different types of activities, and they have different types of definitions based upon how they behave. So we're going to look at isotonic versus isometric contractions. So a contraction event is a force in a length, or a force over a length. If we look at isometric muscle contractions, that's generating tension without changing length. So iso meaning same, metric meaning position. So isometric is you're generating force with no changing length in the muscle. So pushing on a wall, right? Holding a plate of food stationary in two hands or one hand out in the open air. Isometric contraction is maintaining tension or developing tension or producing tension, but you're not changing the length of the muscle. It's straining on the jar of jelly as you're trying to get it open until it actually pops. But until it makes that pop, that's an isometric contraction. Isotonic is very, very different. There's two different types. There's concentric and eccentric. And these pictures at the bottom are here to help illustrate. So isometric, no movement, it's stationary, you're holding it stationary. The muscle develops tension, but it doesn't shorten or it doesn't lengthen. In an isotonic contraction that's concentric, the muscle shortens and the tension remains the same. So isotonic is you've got force generated while shortening the muscle. And it's typically a constant force because you're doing this motion at a smooth rate. And isotonic eccentric is just the opposite, where as the length of the muscle lengthens, you're generating force. Usually, it's a maintenance of the same amount of tension or the same amount of force, and it's like doing negatives with the dumbbells, where you're lowering them and you're generating force. Right? So this gives us all of our daily activities, which are very, very fluid. In fact, in robotics, this is one of the most challenging things to mimic, is the human body's ability for nice fluid motions where they're not jerky motions. So think of any kind of robotics, the little Starlink things that run around campus delivering your food, right? I mean, they kind of like, right? They're on the sidewalk and they're kind of deciding to go or not go. They're kind of doing one of these. So all of us have extremely fluid motions, no matter what it is that you're doing. Okay, So we need to actually flash back to the first unit. Some of you are going to be like, oh, geez, really? Yeah, but it's going to come into play here in a second. And it'll actually make a lot of sense as to why we talked about all this. So we know that muscles, now you can see molecularly, bless you, the rationale as to why muscles need ATP. So share with me why that statement is a true statement. Why do we need ATP for exercising muscles? What's that? Creates energy. But tell me where it's used. I agree, it totally creates energy. Give me some specific examples of where ATP is used. Caden? When the myosin bonds to the actin. Okay? Give me some more specifics there, because you're kind of right. Kind of right. It uses ATP, right, to let go. You need ATP to let go. When it binds, it's not ATP when it binds, right? Because it has to be cocked. So it's ATP here, it cocks, so it's ADP plus PI. Then it binds, and then it uses up the ADP. But ATP is used in that process, I agree. What else? Is that the only place ATP is used in an exercising muscle? What about relaxation? Pump calcium, active transport, pump calcium back into what? Close? Yeah. Sarcoplasmic reticulum, terminal cisternae. Yeah, I know. Transverse tubule and terminal cisternae are very, very close in nomenclature. And they sound like made up words, right? Okay, so ATP is critically important for muscle contraction. So let's look at where we get our ATP from in the muscle. Well, back in the first unit, and back in basic bio, you talked about anaerobic fermentation as well as aerobic respiration. And we're going to talk about it in more detail right now. So in unit 1, we said if you have glycolysis that can generate energy very quickly without oxygen present, the benefits is it's fast energy. The limitation is you only get net how many ATP? 2, not very many. Because if you do aerobic respiration, you actually net 36 plus the 2 from glycolysis. So depending upon how you're counting, it's about 36 to 38 ATP. Much, much more. But it's an oxygen requiring process, and it takes a little longer for it to actually happen because of all of the chemical reactions that need to take place. So we actually have another clever way in the muscle to generate energy even more quickly than glycolysis. And this is using a phosphate donation system. So the phosphate donation system is for quick energy less than 10 seconds. There's 2 places that we can actually strip phosphate groups, which are equivalent to energy. In the muscle, we actually have what we refer to as myokinase. So myokinase is an enzyme because myoglobin takes oxygen into the muscle from the bloodstream, right? And now you have oxygen in the muscle, but you need it for later, and it's not fast, but you have another enzyme known as myokinase, which actually will strip phosphate from other molecules and add them to ADP to make ATP. That's myokinase. You also have another enzyme in muscle known as creatine phosphate. And of the creatine phosphate, the enzyme creatine kinase specifically will do the same thing. You have creatine phosphate in the presence of creatine kinase, creates creatine, takes the phosphate off of creatine phosphate, donates it to ADP in the muscle, and makes ATP. So Creatine phosphate supplies the phosphate. Creatine kinase is the enzyme that strips it off the creatine, delivers it to an ADP that happens to be around, and makes a fully functioning ATP molecule. So these two enzymes exist in exercising muscle and what give us the ability to generate fast energy in the first 10 seconds. So the way this plays out is on this slide. If we have short-term ATP, we call this anaerobic, first 10 seconds of exercise. This is this phosphagen system. This phosphagen system, along with aerobic respiration from the myoglobin that gets started, but really doesn't actually play its effect until after 40 seconds, but it gets started here. But the phosphagen system is what gives us energy for the first 10 seconds of any activity. That phosphagen system is the myokinase and the creatine kinase. And the myoglobin is what strips oxygen off of hemoglobin from the red blood cell, but it's not going to play a role until over here, and we'll talk about that in a second. So the first 10 seconds of activity is from this phosphagen system. Then you actually have glycolysis that takes over between about 10 seconds and 40 seconds. This is the glycogen lactic acid system, anaerobic fermentation, otherwise known as glycolysis. Also fast energy, not as fast as the phosphagen system. But anything over 40 seconds of activity is going to require aerobic respiration, leveraging what we started in the beginning with the activity with myoglobin stripping oxygen from hemoglobin. And this is how you sustain activities that are aerobic in nature after 40 seconds of activity. Now, and it's not like, anybody drive like a manual transmission? Okay, 2 people. So it's not like, I mean, they don't really make them anymore, right? But the system isn't like at 10 seconds, you shift out of phosphagen and you move into glycolysis. And then at 40 seconds, you shift out of glycolysis and you move into aerobic respiration, right? It's a smoother transition. Otherwise, you'd be racing and then be like, right? It'd be like shifting a car. So it's up and then it fades while the other one comes up and fades. And we have this activity, and you watch elite runners, and you can't tell when they're shifting their system, can you? You can tell at the end when they've saved something for the end, right? But you can't tell when they're shifting gears on energy system unless you've ever run into the situation where you're kind of an elite athlete, you're doing something pretty heavy, and you run out of energy or you hit a wall and you bonk. You can tell that something has gone sideways, right? You've actually depleted all the energy stores in the system, and your aerobic system isn't able to keep up. So this is our energy system for exercising muscle. We're tying this into basic bio. When you learned about ATP sources, we're tying it back to the first unit when we kind of refresh where these things come from. And now I'm trying to give you real world examples, real body examples of why it's important to understand where energy comes from. Questions? Generating force, sources of energy, why energy is important for exercising muscle. Okay, now we're going to be weaving a bunch of stuff from the past. So we're going to talk about smooth and cardiac muscle. We've spent the vast majority of this unit until now talking about skeletal muscle. It's the main one. But when we looked at histology and tissues in lab and in lecture, we also talked about 2 different additional types of muscle. We talked about cardiac and we talked about smooth. And we said cardiac is most similar to skeletal, and smooth is the one that's kind of the outlier. So now we're going to get into some of the explanation as to why there are some similarities and why there are some differences. So what do these muscles do for us? Whether it's skeletal, smooth, or cardiac? Well, they contract, they shorten. And when they contract and when they shorten, they perform a function. They have a specialization to allow that function. They have a specialization in their architecture, their microarchitecture. Hopefully, I've convinced you by now that a muscle fiber, otherwise known as a muscle cell, yes, it's just a cell, but it's a very special cell. It's unique. It's organized in a very interesting way, kind of a crazy way. Like the amount of mitochondria that are present, the amount of myofilaments that are present, like actin and myosin, because it needs those things to perform its function. So muscles are cell-rich, lots of cells in them. The cells are the important piece. And they're extremely well vascularized. Extremely well vascularized. So if you injure a muscle, if you tear a muscle, this question came up in office hours today, and I wasn't going to cover it, but somebody asked, so we'll talk a little bit more about it. If you injure a muscle, like you tear a muscle, it's probably one of the fastest injuries to recover from as an athlete because it's extremely vascularized. It'd be way better to tear the muscle and be out for maybe a week or two than to tear a ligament or a tendon. The next best thing is to break a bone, right? I mean, if you got to pick your injury, it's like muscle tear, then bone, right? And these would be on the non-dominant, non-valuable athletic appendage. If you're a soccer player, it's your arms. You want to break or tear those, right? If you're a basketball player, I don't know. I'm not sure what you're going to collarbone. I don't know. It's not a good situation. But you guys get the point, right? Well vascularized, so they heal really well. These are elongated cells or fibers with these cytoskeletal proteins that perform these functions. Okay, so refresh, revisit, looking at skeletal muscle. Now, when I show this image and I say, okay, it's multinucleated, now it means a little bit more because you understand when I looked at this on these diagrams, I could see multinucleated fibers or cells. You can't really appreciate the density of the mitochondria, but you and I both know there's plenty of mitochondria here. You can also see the striated pattern, and now you understand it's because of those actin and myosin thick and thin filaments that overlap. So we say they're under voluntary control. They attach to bones via tendons. They're elongated, multinucleated cells that have a striated appearance. So that definition is the same definition we used earlier in the semester. Hopefully now it actually means even more. We looked at cardiac muscle. These are the same pictures we looked at earlier in the semester. We looked at cardiac muscle. We said it's involuntary. That still should make some sense. It's branched. So, instead of having parallel fibers, every now and then the fibers kind of take a branch architecture. And one of the reasons for that is with skeletal muscles, you'll have the muscle contract in a single plane. With cardiac muscle, when it contracts, it actually contracts as if you're wringing out a towel. So if you take like a washcloth or like a hand towel, not like a bath towel or a swim towel, but like a hand towel or a washcloth, and you get it really, really soaked, and then you squeeze it to wring it, that's the same type of contractile motion that the heart makes versus skeletal muscle, which contracts in a parallel line. But because these are branched, when they contract and pull, they don't contract in one plane. They contract in this plane, but they pull in that plane as well. And so it's more of a wringing motion to squeeze the blood out of the heart. They're uninucleated. They're still striated, so we know the organization mimics skeletal muscle because you see the striations. So the arrangement of actin and myosin and troponin and tropomyosin in cardiac muscle and skeletal muscle are the same. They're absolutely the same. In fact, one of the clinical tests to determine if a patient recently suffered from a cardiac event or a heart attack, any ideas? It's a blood test. It's a troponin lab. So you're looking for cardiac troponin in the bloodstream. Why would cardiac troponin be floating in the bloodstream on a patient just had a heart attack? Any ideas? Cell death. So a heart attack or myocardial infarct, blood flow to the cardiac tissue is ceased for a moment. The cells die. When they die, you get a necrosis event, and the tissue itself is traumatized. If it's traumatized and there's small molecules like troponin, cardiac troponin, it dissociates and ends up in the bloodstream. Cardiac troponin ends up in the bloodstream, and you can actually do a blood draw and test to see if there's any cardiac troponin. There should be no cardiac troponin circulating in the blood unless the heart muscle was damaged. Okay? So you do a troponin lab on a patient, now you know exactly what you're looking for, microarchitecturally. Kind of cool, huh? I mean, not for the patient, but cool from a clinical standpoint. Okay, and the intercalated discs. These are the ones that help make sure that the cells communicate that electrical signal as you ring the heart when you contract to squeeze the blood out. You want to do that in a very organized fashion. So we also saw in the first unit in tissues that there's no nerve stimulus absolutely required for cardiac muscle. So here's one of the places where it differs from skeletal. You have innervation of the heart. You're going to talk more about this next semester. So you can actually ramp up heart rate or you can slow it down using nerve signals, sympathetic or parasympathetic activity. We'll talk about that a little bit in the next section when we talk about the nervous system. But if you took the heart out of the body ex vivo and cut all the nerves and kept it alive, it would beat all on its own. Any Indiana Jones fans? Anybody see the Indiana Jones movies? You know, they found some new recent artifact outside of Petra, and it was like the NPR little speech was about how it looked very, very similar to and was in almost the exact same location in Indiana Jones's Last Crusade. So I don't know. There's something to watch in these movies, right? But the analogy of Indiana Jones, I think it was the Temple of Doom when they took the heart out and it was beating. Like, that's a real thing. Ex vivo, the heart will beat autorhythmically for a period of time. So in cardiac transplant patients, we'll do a cardiac transplant. You don't reconnect the nerves. You actually insert a pacemaker to control the heart because you take heart from one patient to another. But in you and I, the heart has a natural rhythm of 100 beats a minute. Just that's its resting rate, is 100 beats a minute all by itself. That's because it has this pacemaker. And that's nervous tissue intrinsic to the heart that sets the baseline heart rate. Anything below that is parasympathetic innervation that slows it down. Anything above that is sympathetic activity or sympathetic nervous system stimulation that speeds it up. It's aerobic respiration exclusively. Aerobic respiration exclusively, except in extreme conditions like a cardiac event. In a cardiac event, we will look for another element in the blood to give us a confirmatory signal on cardiac troponin to say, did the patient have a heart attack? And that's a lactate dehydrogenase that comes from excessive glycolytic energy utilization. So if the heart is starved of oxygen and can't go through aerobic respiration, it will try its best to keep it alive using glycolysis for as long as it can, which is about how many seconds? 40 seconds, right? Remember that slide? Up to 40 seconds of activity is glycolysis. So for about the first 40 seconds, maybe to a minute, the heart is trying to keep energy alive. After that, the glycolysis fails, and you have this buildup of lactic acid and lactate dehydrogenase, and we'll look for that in the bloodstream as well. Somebody had a cardiac event, they probably weren't trying to run, 40-yard dashes, right? They had a cardiac event, and there's tremendous amount of lactic dehydrogenase along with troponin, cardiac troponin, and those two labs tell you they probably had a heart attack. So the central dogma, that's just a term that means the generally accepted thought in the literature. Central dogma. It's kind of a buzzword you should know. You'll hear it from here on out. Central dogma is the well-accepted main idea, is that cardiac muscle doesn't regenerate. Unlike skeletal muscle, it won't repair. It's well vascularized, but after a cardiac event, it's not going to heal like a skeletal muscle tear. And that's the reason that post-infarct, post-myocardial infarction, post-heart attack, patients have scar tissue on the heart because it actually doesn't repair itself. It doesn't divide. Okay, very, very different. Now we're looking at smooth muscle. Smooth muscle is under involuntary control. Smooth muscle is non-striated. So what does that tell you about the organization of the thick and the thin filaments? Is it the same or is it different than skeletal and cardiac? It's got to be different. Don't know yet how it's different. We're going to talk about that. These are slow, sustained contractions. It's got a single nucleus. These are fusiform. Fusiform means it's kind of like pointy on one side, kind of fat in the middle, and then pointy again here. That's fusiform, or spindle-like. So smooth muscle. What makes smooth muscle so smooth? Well, it's called smooth muscle really because it lacks these striations. It has a smooth appearance. That's one of the reasons we refer to it as smooth muscle. We look at smooth muscle, and it still has Z discs and it lacks striations, it lacks Z discs, and it lacks T tubules. So the organization is very different. It's not quite as rigid as what we see with skeletal and cardiac. It still has calcium. The calcium actually comes from the interstitial fluid. So that's outside of the cells where the calcium comes. It's not stored inside the cell like it is in skeletal and cardiac. And it still has actin and it still has myosin. But the arrangement is extremely unique from skeletal and cardiac. So let's take a peek. Here's the fusiform. See, pointy in the end, kind of fat in the middle, and then pointy at the or at the beginning and at the end. So pointy, thicker, and then pointy. Single nucleus. You can appreciate myosin in a thick filament arrangement. And then you can see the actin filaments that are nearby and overlapping the myosin. But this structure on the left is in a relaxed form, and this structure on the right is in a contracted form, where the thick and the thin filaments slide across each other, and this whole thing just balls up. So it goes from an elongated state to more of a sort of contracted state over on the right side. So these absolutely undergo mitosis. And in fact, in 202, when you look at atherosclerotic disease of the vessels, so the blood vessels have kind of three layers, and the middle layer is made up of smooth muscle on the arterial side of the bloodstream. And as we age, the inner lining, which is the protective lining, the lumen of the blood vessel, is the endothelial lining. It's very thin. And when that endothelial lining erodes or is damaged because of high blood pressure, a lot of pressure on that layer, or contaminants, toxins over a long period of time, lipid-containing blood or high cholesterol blood means the blood is actually a little bit more viscous. It's not as watery. It's more like syrup. And so pushing that through a tube puts more pressure on the endothelium. Anyways, the endothelium compromises, exposing the smooth muscle layer. When that smooth muscle layer is exposed, it actually divides. As it divides, it narrows the vessel. There's other elements like cholesterol and calcium that's in the bloodstream that form lesions, and that's atherosclerosis. So one of the problems with atherosclerosis is that muscle cells can divide, and they can occlude slowly over time. They narrow the vessel down where you don't get blood flow. Okay, continuing on with the smooth muscle differentiation from cardiac and skeletal, we've got two different types here. We've got multi-unit, which is image A on the left, and then we've got single unit, image B on the right. So multi-unit, these muscle fibers receive their own nerve supply. Some examples of multi-unit, image A, the one on the left, are the erector pili, the larger arteries, the pulmonary airways, and the iris of the eye. Now the single unit, these guys all contract in a single unit. So you've got one nerve supply that actually innervates. Here's our nerve supply coming in yellow, and it innervates this entire group of smooth muscle. Most of the smooth muscle in the body is organized as single unit. So they all contract in a single unit. So continuing with some of the differences of smooth muscle, it can contract without a nerve stimulus, just like what happens with cardiac, but it also has autonomic nervous system innervation. That's what the ANS stands for. It has nerve innervation, but it can contract independently through this enteric nervous system that we'll talk about in the next couple of units. It has neurotransmitters, but the neurotransmitters are stored in varicosities, not in vesicles. So these varicosities, you can kind of appreciate, this is the nerve fiber, and it has these little bulges as you go down. These little bulges are these varicosities that house the mitochondria as well as the synaptic vesicles. Then it has, the last analogy are diffuse junctions, which are the synaptic cleft corollary. So these operate just like the synapse, where you get the neurotransmitter being released out of the synaptic vesicle, crossing over that diffuse junction and stimulating an action potential on the muscle side. So how do they work in comparison? So cardiac and skeletal, same kind of activity. Smooth is a little different. We already said it's relaxed on the left, contracted on the right. We still have myosin, we still have actin, but the way that they overlap and the way that they interact is slightly different than what we saw with skeletal and cardiac. But calcium gates are found on the plasma membrane because the calcium supply is outside the cell. So we have calcium gates on the plasma membrane of this muscle cell allowing calcium to come in. These are voltage regulated calcium gates. We also have ligand regulated calcium gates that are impacted or controlled under hormonal control. Because of the vast distribution of smooth muscle across the body, there's hormones that will actually regulate how quickly or how slowly smooth muscle will contract. Lastly, we have mechanically gated calcium gates. So mechanical is, if you get a contraction here, it's going to stimulate a gate next to it to open, allow calcium to flood in, contract here, contract here, contract here. When is that type of motion important for smooth muscle? What's that? Digestion. Just swallowing down the esophagus. That's called peristaltic motion. And it's the same activity that happens during digestion as you move the bolus of food through the GI tract. It just happens a little bit more slowly over like an 8 to 10 hour period, where you get contraction waves coming up, contraction waves coming down. Calmodulin. So there is no troponin in smooth muscle. Instead, we have a structure known as calmodulin. And calmodulin is the troponin analog for smooth muscle. That's what binds the calcium. The calcium binds to the calmodulin, and that's what stimulates the ability or allows for the active sites on actin to be exposed to the myosin head. But this contraction has a long latent period. Long latent period means it has a long period of time between the action potential and the contraction. So you stimulate the smooth muscle with an electrical signal. It actually doesn't do anything. There's a lag for a long period of time. That's that latent period. Okay, it's like when your Wi-Fi is slow. It has a slow contraction, like what we just talked about with digestion. You wouldn't want to contract smooth muscle too quickly and have the bolus of food move through you too fast because you wouldn't be able to absorb the nutrition. And it also has a slow relaxation as well as slow contraction. So, lots of differences between skeletal and cardiac as well as smooth. A lot of unique idiosyncrasies. Skeletal and cardiac behave more similarly with the way they contract. Smooth muscle has its kind of own unique way of contracting. But if you compare and contrast skeletal, cardiac, and smooth, you'll see some unique differences between the three of them. Bless you. Okay, so here's a question for you. A couple of questions where we move into the final section of today and the final section of Muscle Phys. In 1997, Wilson Kipker of Denmark set the world record for the 800-meter dash, 1 minute, 42.67 seconds. And as he was finishing his race, which energy source predominated in fueling his leg muscles? So what energy source was happening towards the end? Raise your hand if you want to answer this one. C, aerobically generated ATP. Why? It's longer than 40 seconds. Well done. Okay, this contraction is an example of what? A, isotonic and concentric. Agreed. Why? The bicep is shortening, so tension is being produced while the muscle is shortening. Okay, how about this one? This picture is an example of blank contraction. C, isometric. Why? Someone in the back. Tension in the muscle, but it's not moving. Hopefully it's not moving, right? Otherwise your food's going everywhere. Okay. All right. How about this one? This picture is an example of what type of contraction? First off, what muscle group is being exercised? I know this is more lab, but what muscle group is being exercised with this? Triceps, right? Primarily triceps, not exclusively, but that's okay. Which one is it? B? Why? The muscle is lengthening and generating tension. Okay, fantastic. All right, in the last remaining time, we're going to talk about different types of muscle fibers. And then I got a few more questions at the end. So as we look at the different types of muscle fibers, it's kind of fascinating because the muscle itself isn't made up of just a single type of muscle fiber, even though in the diagrams, we've really just been describing muscle fiber as a muscle fiber. We have different varieties, some that prefer glycolysis and others that prefer aerobic respiration. And there's no muscle in the body that's pure glycolysis, like the glycolytic muscle. Every muscle is made up of different types of muscle fibers, but they could have a higher percentage of one type over another because they're more geared toward one type of activity versus another. Now, in the example of athletes, there's some training specificity here too. So depending upon what kind of training the athlete is doing, you may preferentially encourage the development of one fiber over another. So for example, if your training regime is a lot of 10-second spurts of energy, 10 to 40-second sprints, interval training, quick bursts of energy, you're going to favor a fiber that likes glycolysis. As you break down that muscle tissue and you regenerate and rebuild, you have hypertrophy of the muscle, that's enlargement of the muscle. You have hyperplasia of the skeletal muscle, where you build new muscle fibers. You're going to lay down muscle fibers based upon the demands of the activity that you're placing it under. Does that make sense? This is why your coaches are telling you, it's not just practice makes perfect. It's what? Perfect practice makes perfect. You need to practice as if it's the race, because that's what you're training for. And so the muscles do the exact same thing. So the first type of muscle is slow muscle fibers. Has a couple of other names, depending upon the literature. Be called red muscle. It could be called slow oxidative muscle. They are called type 1 fibers in certain literature. In this muscle histology picture, a cross-section through a muscle, you can see the slow oxidative fibers are kind of the darker red. They almost look brown. Depending upon your image, they may look black, like black round circles, right? Because of the high amounts of mitochondria to run through oxidative phosphorylation and use aerobic sources of energy, they have a very dark red appearance. So they're slow, long twitch. They have less power because they have a slow myosin isoform that develops in them. They're not made for fast contraction. They have more myoglobin, and they have higher density of capillaries than the fast glycolytic ones that we're going to look at. Why? Why do you have more myoglobin and more capillary density in slow oxidative muscle fibers? Any ideas? What's their energy? Go ahead. They're staying contraction for longer, and they're using what source of energy? Aerobic respiration, so they need oxygen. If they need oxygen, they're going to have more myoglobin to take the oxygen off of hemoglobin, and they're going to have more capillaries to deliver more oxygenated blood to them. Make sense? Postural muscles, the ones that you use in this classroom for an hour and 15 minutes twice a week, keep you sitting upright, right? Those are examples. Fast muscle fibers, the FG in this picture. And you can see that this prep is favoring fast glycolytic fibers. So these are fast, short twitch. They're usually larger in size than slow. A couple of different names. They're called white muscle, fast glycolytic fibers, or type 2 fibers in the literature. High power, but they fatigue very quickly. They have a high concentration of creatine phosphate in the glycogen lactic acid pathways because they do a lot of phosphate borrowing, because these are fast muscles for the first 10 to 40 seconds. Now, all muscles are going to have both of these fibers, right? An example of the fast muscle fibers would be like the gastrocnemius muscle. Now, all muscles are going to have both. So if you look at kind of a slice of a population here, you can see slow oxidative in this first column, fast glycolytic in the far right. And you can kind of see this is focused on male athletes. Sorry, ladies. It's pretty biased, but that's what they were looking at. Marathon runners, swimmers, average individuals that don't have a specific training regime that they focus on, and then sprinters and jumpers. So if you look at marathon runners, 82% in the quads, right? The quadris femoris group, that's basically your thigh muscles. 82% of marathon runners of marathon runners that were surveyed, 82% of their quadriceps femoris group was slow oxidative fibers. That should make sense, right? They got to run a long race for a long period of time using oxygen primarily as their fuel source. Swimmers, 74-26. Now look at kind of the average Joe, almost 50-50 in the quads. Because some days you got to run away from a bear, other days, right, you got to run all the way home because your car broke down, right? That's the average Joe's life. Sprinters and jumpers, more about fast glycolytic, fast bursts of energy, right? Training specificity, though, too. Like, this isn't set in stone. Any one of us in this room could be put on an athletic training program. We could take a muscle biopsy of the quadriceps before and after training, and we could shift your percentages, every single one of us. Some of us more than others. And some of us start genetically already favoring 1% over another, right? It's kind of interesting how that works. All right. Muscle cells enlarge. That's the whole point of this picture. It's not to try to cast if this is beautiful or not beautiful. It's just to bring home a point that you put muscles under stress, they will enlarge significantly. Red muscle adds more mitochondria and myoglobin. And if you put muscles under pressure, like stress and tension, not just skeletal muscle, but cardiac muscle, you'll actually enlarge cardiac muscle the same way. So this is the reason that you have these cardiac hypertrophies that take place in a lot of athletes that are over-exercising, or in athletes that are maybe supplementing in ways that they really shouldn't be supplementing, right? And we see that. Like, some of the best examples are some of the bodybuilders that came out of the 70s and the 80s, like, Arnold Schwarzenegger himself. He's actually not, he's pretty vocal about it, and it's in the public domain. He's had quite a few cardiac surgeries to actually do a heart reduction, where they literally come in, cut a section of the heart out, and then sew it back together to try to reduce the size of the heart. And the Governators had a few of those. Okay, why do we fatigue? Why do muscles wear out? Now, this is different than psychological fatigue, right? So if you're an athlete, and I know this is very athletically pushed, but sort of one of the better examples of how skeletal muscle works is looking at athletics, okay, or exercise. So I know in athletic performance that, there's lots of manuscripts, a lot of papers that talk about, fatigue is mostly here, right? It's psychological. Now, I'm talking about the 10% that is physiological and not psychological. There is a physiological deficit that can take place. So ignoring, the mental game, all in your head, as Coach says, and he's not or she's not wrong, but there is a contribution that's physiologic. There's many possible explanations here of why we burn out from our muscles. We can get leakage of the calcium into the sarcoplasm. We run out of energy. No more glucose, no more glycogen stores. ATP shortage, right? Breathing can't keep up. Aerobic deficit. The release of potassium lowers the excitability of the membrane. So we saw in excitation-contraction coupling that sodium rushes in and potassium leaves. Now that membrane is less excitable the next cycle around. You can reset those ions using the sodium-potassium ATPase pump, but that also takes energy. So if you don't have enough ATP, you can't reestablish that membrane potential for the next contraction event to take place. What about neurotransmitter cycling? Right? Neurotransmitters are critically important. Change in pH? Probably not. So there's a lot of theories around lactic acid buildup in muscles and them leading to fatigue. Typically, your buffer system in your body physiologically can get rid of that acid burden pretty quickly. In fact, what we blow off as CO2 and water is from a buffer system in our bloodstream known as the bicarbonate buffer system. So we have bicarbonate that floats around in the bloodstream. You'll learn about this more in 202. And in the presence of free hydrogen ion, which is what comes out of an acid burn from muscles, it actually gets buffered by the bicarb and turns into water and CO2. And that's what you breathe on a mirror and you'll see water vapor, and you'll collect CO2 if you have a CO2 meter. So we can buffer pretty quickly. That's why when you exercise, your respirations go up, not just to bring more in oxygen, but to get rid of more acid. Because you're building up a lot of acid in the muscles as you exercise, and so you blow it off in the form of buffered acid. So to that point, after the event is over, you finish running a race, you're still huffing and puffing for a period of time. It's not like your respirations immediately go back to resting. And that's because you still have this deficit. This lactic acid needs to leave. It needs to be buffered. It'll move to pyruvic acid. Most of it's converted back into glucose in the liver. But you also have to replace all these oxygen deficits from the exercise, reestablish myoglobin in the muscle and hemoglobin in the blood, and then replenish any of the creatine phosphate that you used during the activity. All of this is requiring energy, and the energy needs aerobic sources. That's why the breathing rate is still high after you exercise for a period of time. Last but not least, every time you strip one of those phosphate groups off to make ATP, you actually need to donate them back. Okay. When a muscle's at rest, which of the following is not chemically associated with the other? When a muscle is at rest, which of the following is not chemically associated with the other? Do you understand the question? Okay. Any takers? Myosin. At rest, myosin is not bound to the binding site on actin. Okay. A great exercise for you to take a picture, be able to label this image. This is what? Sarcomere, right? Showing a sarcomere. Another picture. Label from a muscle cell. Muscle fiber, also known as a myocyte. Questions? Okay, that concludes muscle physiology, but stay tuned for a second.